Okay, now that we've learned some enzyme basics in terms of jargon and vocabulary and mechanisms, let's look at the properties of these miracle proteins. First of all, write in your notes that enzymes are reaction specific, which means that each enzyme works with a specific substrate. Also write that they accomplish this with a complementary chemical fit between the active site on the enzyme and its specific substrate, and that this is accomplished using both hydrogen and ionic bonds. Also add that enzymes are not used up or changed in the specific reaction they catalyze, meaning that once the reaction's done, the enzyme is free to move on to the next substrate and catalyze it, and then repeat the process over and over again. The special thing about these proteins is that they are unaffected by the reaction they catalyze. Now, lastly, write in your notes that the rate at which enzymes work is influenced greatly by the conditions in which they work. For example, the temperature, the pH, or say the salinity of the environment. In an earlier podcast, we discussed how enzymes get their names, but it's worthwhile repeating it now that we're actually talking in detail about them. Write in your notes that enzymes are named for the reaction they catalyze and the substrate they work with, and most times have an ASE ending. For example, you see sucrase here. So sucrase now, you see sucrase. Here's our substrate. Okay? You see sucrase, which would be the enzyme that breaks down sucrose. So here's sucrose. Sucrase would be the enzyme now that works with sucrose. Now, proteases break down proteins, and lipases break down lipids. Now, we've also seen through our study of protein synthesis a molecule called DNA polymerase. You see it right here, DNA polymerase, which adds nucleotides to the DNA strand, and RNA polymerase that copies DNA and produces messenger RNA. Those are all examples you should have in your notes with an accompanying explanation. Lastly, we see pepsin. Here's pepsin, no ASE ending here. Pepsin, which breaks down the peptide bonds in proteins. You'll need to know examples for the AP exam, so pick a few of these and get them in their notes if you need to. Okay, now back to the lock and key model we introduced earlier. Here you see a much better example of how this model works. Let's get the 4 basic steps in this process in your notes, and if you want to, you can draw this little circle diagram here with the arrows. That's probably a good idea, but whatever you think is going to fit your needs, you're going to need to draw this and get the 4 steps in that we're going to go over. All right, let's talk about step 1 first. Step 1 is where the substrate, in this case sucrose, so here's our substrate, and we've already talked about sucrose in a podcast. You already know that sucrose is made up of the monomers fructose and glucose. So here's fructose and glucose right down here. So sucrose now is our substrate. Now this binds to the enzyme, in this case sucrase, at the active site. So now you see in step 1 sucrose binding now to the active site of the enzyme, creating this enzyme substrate complex. Question: what kind of a reaction is this? If you said hydrolysis or catabolic, you'd be correct. If that's true, then somewhere in this diagram we should see a water molecule of some kind being added to this reaction, right? Well, here it is. Here's the water molecule right here. Now, in step 2, the substrate is converted into product which in this case are fructose, which is acting like a Bronsted-Lowry acid. Why? Because it's accepted a hydrogen ion. So here's fructose accepting that hydrogen ion, so it's a Bronsted-Lowry acid. Whereas glucose has accepted this hydroxide molecule and therefore is acting like a Bronsted-Lowry base. In step 3, the products are released from the enzyme. So you see that here, the products are released from the enzyme in step 3. And then in step 4 now, the active site is now available for the next sucrose molecule. So again, let's review now what's unique about enzymes. Enzymes do their job, i.e. reduce activation energy and speed up reactions, but themselves aren't used up or changed. So again, this substrate moves right into the active site. This reaction takes place, this hydrolysis, with the addition of water. Boom, here's our products. And now the enzyme is unchanged. It's ready now for another sucrose molecule. So that's the beauty of enzymes, is they're not used up. They just keep doing their job. Whatever specificity it has, they're ready to do it again very soon. This reaction happens extremely fast. And I'll look at that when we do our next series of labs on enzymes. Now let's be even more specific with our model of this mechanism called the induced fit model. Write in your notes that the induced fit model is a more accurate model of enzyme action, and that in this model the binding site of the substrate actually causes the enzyme to change shape in order to create a tighter fit with its substrate. So again, you can see the shape of the active site here. This is kind of a general shape, but when that substrate fits in here, you can see now this molecule close in, and we're going to talk about that in our labs as well. The first lab we're going to do is a toothpick lab. You might remember if you had me for regular biology. And we'll see now where your fingers now function to close over that toothpick. So again, that's called a conformational change. You see the word form in here. So literally what happens is this enzyme conforms to the shape of the substrate. You need to get that in your notes. That's called the induced fit model. So literally the substrate induces the enzyme to change its shape to fit the substrate. Well, of course we need to know how this works, right? Two mechanisms we need you to get in your notes. First is synthesis or anabolic reactions. Write in your notes that it's these reactions where the active site actually directs or orients substrates into the correct position to bind with the substrate. In effect, the enzyme brings the substrate in closer to the binding site. Secondly, in digestion or catabolic reactions, the active site actually binds the substrate and puts stress on the bonds that have to be broken, making it easier for that molecule to separate. So make sure you get those two mechanisms in your notes. Now earlier we mentioned that enzymes do their work without being used up or altered in any way. But we also mentioned that they can be affected by changes in environment, like temperature, pH, etc. So when we say that enzymes do their job without being altered, we're assuming that they are performing in an optimum or perfect environment. Write that down. For example, in your body, the temperature is 98.6. That's the perfect environment for the enzymes in our bodies to work. However, we also know that that environment can change due to injury, illness, etc. So let's look at how a change in environment can affect our friends, the enzymes. Here's a list of the factors we're going to take a look at some point in time. Can you guess why the list is so long? Yes, the list is so long because at one time all of these have appeared on the AP exam at one time or another. So let's get some of them in your notes now and get a brief description of each. By the way, this will make an excellent addition to your flashcards as well. First, let's look at temperature. It should be no surprise that enzymes function optimally at certain temperatures. Write in your notes that enzymes are inactivated or denatured, meaning by temperature above 50 to 60 degrees Celsius. Also write that enzymes are not inactivated by freezing, but either work slowly or do not work at all when the temperature is too low. Also write that only the hydrogen bonds are broken at normal temperatures. In order to break the primary structure, i.e., for example, the peptide bonds, you would need to boil these proteins in strong acid for several hours. Not good. Or simply use the protease enzyme. Well, that makes more sense. Okay, now for pH. Write that enzymes are sensitive to changes in acidity. For example, the high acidity of the stomach juices is needed for the enzyme pepsin to function, whereas an enzyme called trypsin in the small intestine requires an alkaline or basic surrounding. Okay, now concentration of enzymes. Write that if pH and temperature are kept constant, the rate of reaction, and this is important because we're going to expound on this, the rate of reaction is proportional to the amount of enzyme present, and that the more enzymes there is, the more reactions that occur. And we're going to turn that into a rate here very quickly. Then there's coenzymes. Write that some vitamins and minerals, like copper, zinc, and iron, in small quantities work with enzymes to speed up reactions. It's why most physicians and nutritionists suggest that you take a high-quality multivitamin every day, which I do, by the way. All right, now for enzyme inhibitors. A pesticide called DDT stops the actions of enzymes in insects, thereby killing them. Now, we're going to talk about enzyme inhibitors in some more detail later on in other podcasts. So if there's inhibitors, you also see the word activators. So those would be the opposite of inhibitors. So again, you're probably not surprised that enzymes are complicated molecules. Okay, now let's look at reaction rates. In other words, how do some of the factors we just mentioned affect how slow or fast an enzyme can do its job? First, get this graph in your notes and make sure to label it correctly. We're going to use it several times in this podcast and then also in our upcoming labs. Look at the curve that's created in this graph. Can you explain the relationship between enzyme concentration and reaction rate just by looking at this graph? Well, that's where we're going to start part 4. So this is the end of part 3. Take a picture of your notes, submit them to Moodle, grab that study guide, look at those questions, and answer them, and then submit them when you're done. And we will see you in part 4 of our discussion of enzymes. Bye.